Chapter 14, Section 2. We continue to look at genetic information and how is that passed on from parents to offspring, and what can we learn from this information. We're going to backtrack just a little bit to discuss some of the genetic information that is actually located on chromosomes. So we know chromosomes are made up of DNA, all of our genetic information, but only 2% of the DNA in your chromosomes are actually going to function as genes. So those are the things that are actually going to code for those specific traits that are passed on. So you have large portions of your DNA that aren't going to code for specific proteins. Your average gene consists of about 3,000 base pairs, and we haven't touched on this yet because it's in Chapter 12, but if you remember probably from your early grade school days about what DNA looks like. So we have this double helix, and throughout the double helix, kind of this latter piece are these base pairings. And there's 4: adenine, thymine, cytosine, and guanine, and we will talk more about those in Chapter 14. But recognizing there's about 3,000 of them in an average gene. But genes can be larger, genes can be smaller. The largest gene in the human has over 2 million base pair. So there's quite a variation in that. So what can we learn from different chromosomes? Well, again, as our scientific knowledge increases and our ability to research and understand things, we've begun to pinpoint where are specific traits located on individual chromosomes, and are there specific genetic disorders that we can find on these chromosomes. So we're going to look at 2 of them, and these were kind of the first ones that we researched. So we actually know the genetic sequencing for these chromosomes, and on chromosome 21 there's about 32 million base pairs, so average, a little bit higher than average, and there are 225 different genes that are on this chromosome. There are many portions of this gene that doesn't have any genes at all. They're not coding for specific traits. And one of the genetic disorders that we find on chromosome 21 specifically is Lou Gehrig's disease, which is ALS. And this causes progressive loss of muscle control due to the destruction of nerves in the brain and spinal cord. So we know that that is on that chromosome. Chromosome 22 has 43 million base pairs and 545 different genes that are located on that chromosome. There's 2 specific genetic disorders that are related to this chromosome: leukemia and neurofibromatosis. And both of these are kind of that cancer. The interesting part about chromosome 22 is the fact that there are these kind of long stretches where there's just repetitive DNA. They don't code for specific proteins, and these long stretches are unstable sites where rearrangement can occur, and therefore you get if you get this rearrangement, the possibilities for mutation are there. So we talked a little bit about 2 other chromosomes. So let's look at chromosome pair 23. These are the sex-linked chromosome. Sex-linked genes are found on the X and Y chromosome. They're found on the sex chromosomes and therefore is known as sex-linked genes. There are more than 100 sex-linked disorders that have been mapped to the X chromosome. In comparison, the Y chromosome is much smaller and only contains a few genes, so a lot of the sex-linked disorders and whatnot are going to be related to that X chromosome instead of the Y. So here's a little visual of just kind of the X and Y chromosome. Reminder, males just have 1 X chromosome and 1 Y chromosome, and females have 2 X chromosomes. All the X-linked alleles are expressed in males even if the trait is recessive, and that has to do with the fact that there's very few genes or genetic traits be transferred on that Y chromosome in comparison to the X chromosome. This is the stuff that, again, is just going to be this little bit of review, but involving the Punnett square and the sex-linked genes. So let's say, for instance, the mother was a carrier, and we're just going to write an X and a C and an X and a lowercase c. So the dominant would be the capital C, lowercase recessive would be the smaller case, the red C. So if we look on here, the mother was a carrier, so her offspring when she's paired with a male, and if he is not a carrier, he's going to have the capital C and a Y. And again, the big piece about this is the male is going to either express the gene or not. He can't actually be a carrier. It's either going to be expressed or it's not. So the option is 50% females and 50% males. And of these, you have the possibility of a female getting it and the possibility of a male getting it. 50% of the daughters will be a carrier and 50% of the sons will be colorblind. So red-green colorblindness is found in 1 of 10 males and only 1 in 100 females. So there can be some variation with that. But this is what we're going to be practicing is how do we do Punnett squares for sex-linked traits or alleles. Again, we can use the pedigree that you've used earlier in the chapter to kind of look at the passage of these traits on. So we look at the first generation here where, again, the circle is the female, the square is the male. And passing this on, we have a male getting it and another male getting it. And what we see here is that the male here, you don't have a lot of information from this. And when we go on further, we can kind of see some other information. But notice the trends here is where we get the predominantly of the males that are going to see that gene and no specific expression later on. So again, using your pedigree, you can kind of find more information on that. We will come back to this one later. There's a specific disorders, again, associated with the sex-linked genes, and one of those is hemophilia. Hemophilia is clotting of a blood controlled by 2 separate genes. A recessive allele in either of these 2 genes causes hemophilia. And again, hemophilia, for those that you aren't familiar with, it's the inability to clot blood. So 1 in 10,000 males are born with hemophilia, but there is treatment for it. So now there can be an injection that'll actually help them with clotting of those proteins. So hemophilia is one of those. And if we just look at some of the photos here, we can see we have this clotting issue. So if there's some type of injury, they bump their knee, run into something, there is massive amounts of kind of blood pooling because their body can't clot for it. So again, if we go back here, the issue is because the recessive allele in either of these 2 genes causes hemophilia, that's why males are kind of more highly affected than females. We also have Duchenne muscular dystrophy, and this is another sex-linked disorder that results in the progressive weakening and loss of skeletal muscle. 1 in 3,000 males are born with this specific disorder. And it's caused by a defective version of the gene that codes for the muscle protein. And again, the nice thing about understanding kind of how this is passed on is they are spending lots of time trying to research to either treat or cure. And we'll talk more about this in later in the chapter about what kind of things can they do when we know there's a specific allele that is causing this mutation. And we know that this is on the 23rd chromosomes, so we have that information. Another thing that can happen with our sex-linked chromosomes is something called X chromosome inactivation. And basically, when there's 2 X chromosomes that are present in each cell, one is randomly switched off. The turned off chromosome is known as a bar body, and basically it just forms a dense region in the nucleus. So if this focuses on 2 X chromosomes, why is it not found in male? Well, reminder, males only have 1 X chromosomes while females have 2 X's. And that's why we're going to see this more we're going to see this in the females versus the male. It's found in many mammals. In cats, the gene for color coats is located on the X chromosome. So as a result, female can have a more variety of their spot color than males based on this inactivation. There are other chromosomal disorders called non-disjunction. And non-disjunction literally means not coming apart. And if we think back to meiosis, how they're supposed to separate, something goes wrong. And this isn't specifically with the sex-linked genes. This can be all of them. This results in more than one of an individual chromosome. So let's kind of review here. If we look back to our meiosis, I'm going to move this up just a little bit. And notice again, we have our tetrad forming, they're coming in the middle, and then something happens. They don't separate how they're supposed to separate. So what happens is we go into meiosis 2, all of those stages, and we get this unequal amounts of the chromosomes. Because we have this abnormal number of chromosomes, they find their way into their gametes, and then we have different disorders. So 3 that we'll kind of look at that are in your book are Down syndrome, Turner syndrome, and Klinefelter's syndrome. Down syndrome is when you have 2 copies of an autosomal chromosome that fail to separate. So they have 3. It's also called a trisomy. In the United States, 1 in 800 are born with Down syndrome. Trisomy 21 is the most common form. There are other forms in there. But if we look again at our karyotypes, our images of our chromosomes, we can see here that we end up with 3 for the 21. Turner syndrome occurs when a female only inherits a single X chromosome. So for some reason, this would be Turner syndrome would be related to would be a sex linked. We only get that one X chromosome, and because of that, they are sterile, meaning they cannot reproduce, and their sex organs have not developed. Klinefelter syndrome occurs in males, and it's a non-disjunction where they get 2 X chromosomes along with a Y chromosome. So you're going to see it as a 47 and then 2 X's and a Y. And again, just like in the other one, it causes sterility due to the interfering with meiosis, and some have actually been found to have up to 4 X chromosomes. So again, there's some issues here with the separation that's supposed to take place in meiosis. If that is not caught and fixed before the gametes separate, we can run into some issues. The last 2 pieces on here, again, the whole point of this is just gathering information and then, okay, what can we do with this information now? That's what scientists, the medical profession has been looking at, is now that we're starting to understand where genes are located, what can we do about it? Well, one of the things that they've been working on is this idea of gene therapy. Gene therapy is a process used by scientists to replace a faulty gene with a normal working one. And what they do is they insert the gene into a piece of viral DNA and basically begin infecting the body. And what that does is it allows those proteins to function normally. So all of these kind of conditions that we've been talking about where there's been a mutation and something's not happening correctly with the protein and its function, this goes in and kind of helps prevent that or fix that. But it is not 100% effective in all disorders. And again, as our scientific knowledge and experimentational ability increases, and as we learned where more and more of these mutations are occurring, this can only improve. The last little piece is, again, DNA fingerprinting. DNA fingerprinting can be used to determine whether blood or other body fluids left on a crime scene matches the DNA from a suspect. And what it does, it looks at sections of DNA that vary wildly from one individual to another. So because our genetic DNA and our sequencing is so different, that they can use this, get a piece of it, code it, sequence it, and determine how close is this to a potential suspect. Obviously, we've all seen those crime dramas where there's some issues and stuff with that, but that's just another piece of that puzzle. Here ends the notes.